All right. So first, acknowledgement of country. I start by acknowledging the Berigal and Gadigal people who are the traditional custodians of the unceded land on which my lab at UNSW stands, and pay my respects to elders past and present. Also welcome all indigenous people joining the seminar for today. All right, so just for context, my lab does quite a few things. Today you will hear these two stories. The first is called HyperSeq, that's the micro adventure, and then the second is called Cell Atlas Approximations, or just approximations, that's the macro adventure. So I've been told that Yue, who has seen the approximations already, is bored by all things. So I specifically dedicated 80% of the time to adventure one, so she don't complain. All right, the main reason why we are looking at cells is because they're beautiful, obviously. That doesn't need to be said in this room. I know that's your motivation as well. And this is an example thereof, realized or made by this company that happens to also sell microscopes that make beautiful images. And do you know what cell this is?
Barry.
Epithelial cell.
Yeah, blood vessel, not epithelial, but good news. All right, now cells are also diverse. They sometimes fuse together like muscles. They can be small and have little hairs, or have liquid droplets, or be very flat, or have long arms. Or they can be, well, yeah, so they can be, yeah, so there is that. Do you know what this one is? Plant roots. Okay, you see they have cell wall, they're square, and so on. Anyway, very diverse, right? And that's what makes it interesting, right? Not unlike human diversity, right? In which there are all kinds of people, black and white and colorful, and tall and short and whatnot. And that's what makes it interesting, right? Same story. All right, now, diversity has been studied for many decades in biology in terms of organisms, right? So maybe there's something we can learn from evolutionary biology and apply it to cell biology. And if you look at how people started to characterize the kind of diversity of species or organisms on Earth, for a long time, people just did these little sketches or photographs of all kinds of whatever this is, birds. And there are all these, like, bearded men that go on ships and they make sketches and they decide that these two things look similar, therefore they must be related. And that's fair. I mean, and also, to be fair, it's very accessible, right? Everybody can do it, so it's great. However, of course, there is this thing called mimicry. So sometimes organisms look similar, but they are unrelated because they just want to look similar for whatever purposes. And so when nucleotide sequencing came about, and protein sequencing for that matter, people started sequencing conserved genes from all kinds of organisms and realized that that's a more reliable way, if a little more technical, or perhaps a lot more technical, to understand the relationships between organisms, right? And at the top right, you can see a little bit of magic, more or less, right? Because it so happened that there is a mathematical structure that can summarize a lot of evolutionary biology all at once in one global model, the phylogenetic tree or tree of life, right? And you're double lucky because it just happens to be a model that is incredibly easy to visualize and to understand. That's where you can see it. All right. Back to cell biology. Is there anything we can learn from this? Well, if you think about what people have done to characterize cells for decades, basically it's the same thing. They take a lot of pictures. You need microscopes because they're small, but it doesn't change, the fundamental method. And then at some point, thanks to things like flow cytometry and single-cell sequencing later on, it became possible to use essentially molecular counts as a way to characterize and profile cells. And that's how we are nowadays looking, by and large, at cellular diversity, right? Unfortunately, we're not that lucky. You can make trees or hierarchical clustering on cells, and it just doesn't work very well. In fact, no one does it. What everyone does instead, they do non-hierarchical clustering, and it can be done either by hand. Fax people call it gate, or you can do it with graph unbiased, what's it called, unsupervised graph clustering algorithm like Louvain or Leiden or whatever else. And that's the clustering. You still cannot look at it, right? So to look at it, people have developed another set of algorithms called embeddings, and UMAP is one example of that. So it's not quite as convenient, but in a way reminiscent of what happened in evolutionary biology nonetheless. All right, end of the intro. Story one is about putting together these two things: morphology of cells and generally optical appearance of cells and molecular counts. And this was done by Dr. Riquechier, who was a PhD student in my lab until mid last year. She's back from Europe today. So if you have questions, you can ask her. So the idea is to combine information from imaging or high content imaging and omics from the same cell at some reasonable scale. And just to dispel any doubts, we're not trying to do what spatial transcriptomics does. So we're not trying to figure out the location of the cell compared to other cells. We're not trying to figure out the location of the RNA molecules compared to the cell. We are trying to do other things, for instance, looking at cell size, shape, color, subcellular features, potentially organelles like mitochondria, nucleus, etc., etc. And we do on the omics side gene expression in this paper because it's a proof of concept and it's easy, but you could do ATAC-seq or methylomics or whatever else. And the work is done together with Eva Goldis, who is a professor at biomedical engineering at UNSW. We looked at cancer cell line, FCF7. It's a very established old cancer cell line. Just again, this is a proof of concept for the method, right? But we have done the same on primary cells. We've done it on all kinds of different things, and it works, it's robust. And the way you combine optical information with omics from the same cell is the following. So you first need to do the imaging because sequencing kills the cell and destroys it. So you can do that first. So you do the imaging. In this case, we use a special microscope called hyperspectral microscope. More on that later, but you can use whatever microscope you like. Then you take the cells and you move them to a different building, a different floor, whatever you need to do, to another machine that we have, which is called the cell selector, and it's a cell picker. Or like Jean likes to say, a cell pusher. It's the same. Was it pusher? So the second machine picks the cell and puts it in a PCR plate, and then you can apply normal libraries like SMART-seq2, SMART-seq3, ATAC-seq, whatever you want. And then you sequence, and then you do the bioinformatics. And the bioinformatics will be all about connecting the optical features, which are quantitative, with the genomic features, which are also quantitative. All right, the first thing we look at is cell size. Again, this is a cancer cell line, the most boring thing on Earth. And when you look at small cells and big cells, so here the cells are colored with fake blue, just to make you understand a little bit where they are. You notice that the largest cells are around 8 times bigger than the smallest cells. And that's more than you expect, because in a cell line that is dividing, you expect a factor 2, right? You expect cells that just divided to be half the size of cells that are about to divide. That's just the scaling law, right? The geometry. But these ones are 8 times larger. So even within the most boring thing, homogeneous thing on Earth, which is a cancer cell line that's 40 years old, you have an excess of heterogeneity no matter where you look, in this case at cell size. So of course, then the next thing you can do with this, because you have from the same cell both imaging information, but also transcriptomics, is you to correlate the two. And when you compute the correlation, you get a bell-shaped curve like the one you see on the left. And most genes are in the middle, right? Like most genes are expressed at the same level, no matter if it's a small cell or a big cell, that's expected, but you do have tails. And so there are a bunch of genes in the left tail, meaning that they are downregulated in big cells, and you have a bunch of genes at the right tail that are upregulated in big cells. And on the right, you can see a few examples of genes that are anti-correlated with cell size, not correlated, or positively correlated. Now, interestingly, especially if you look at the downregulated genes, you find a lot of members of the spliceosome. That was very surprising to us, but it's a very robust signal. So then the question is, can it be that big cells and small cells splice differently? Okay, So to test that, we went back to the reads, and what people normally do with RNA-seq is they take reads from all kinds of exons within a gene and sum them all up and say this is the number of reads for that gene. And that's fine, but what you can also do is you can take the reads separately for each exon and then check if cells that are, say, big or small, use different exons. And that's what we've done here. And so basically, CS is an example of a gene, but there are many, as you'll see in a second, where even though the gene is evenly expressed across cell sizes, so small cells express as much as big cells, the actual exons that are being used are not the same. So small cells really like exon 1, so start the transcription a little early, whereas big cells really like exon 11, meaning that they start later and then they also end later. What does this mean? We don't really know, but what we do know is that there are hundreds and hundreds of genes just in this cell line in which this happens. So you have the smallest correlation, the exon with the smallest correlation of the x-axis and the highest correlation on the y-axis. The fact that this isn't very much around 0 tells you that it's quite routine for genes to fine-tune their splice-independent size. All right, the other thing you can look at in addition to morphology and bright field images is the colors, and that's where the hyperspectral or HyperSeq comes into play. So these microscopes from Eva Goldie's lab are very special because they can collect autofluorescence signal from the cell, meaning we don't have GFP or anything like that. We just look at the natural fluorescence of biomolecules inside living cells, and they have very, very sensitive cameras to do that. And they also have very sensitive optical filters. So you don't just have 3 fluorescent channels, you have in this case 15. You can have many more. There are setups out there with 200 channels. And this is pixel-wise, right? So for every pixel in your image, you have 15 numbers that tell you the fluorescence in each one of those channels, plus the bright field. And in the middle, you can see, you know, the image of the same cell in all different channels. And the simplest thing you can do if you don't want to deal with any kind of mess is you can average all the intensity for each channel through from the entire cell. And that's what you see on the right. So every cell becomes a 15-dimensional vector of positive numbers. And we call it the cell spectrum. It ignores any spatial heterogeneity within the cell, but it's a simple way to look at things. You don't have to do this, though. If you wanted, you could also look at the individual pixels or other things. And after you do that, you take all your cells, you run PCA on these spectra, and you find out that out of the 15 channels, they roughly group in 2 channels, PC1 and PC2. And some channels have very high loadings in one channel, some in the other. And then you can scatter your cells along those 2 PCs. And indeed, you can then correlate that with gene expression, and you find that there are a bunch of genes that are especially highly expressed by cells that also have an optical PC1 component or PC2 component, and so on and so forth. You're not limited to PCs. You can do it on individual channels. You can do it on subcellular features, and so on and so forth. But this is just one example of it. If you look up what those genes do, those genes have nothing to do with spliceosome. They have nothing to do with cell cycle. Most of them have to do with cellular metabolism and energy consumption. So what does that mean? Again, this is a very exploratory study, but I think it's fascinating that you can take a bunch of cells from the cell line and look at them in this extreme detail to find what the diversity means. I will do more things in the paper. The preprint is out. We find really weird hyperspectral rings at the cell membrane. We find subcellular structures that we don't even look at. You can look at specific optical channels that overlap with the resonant peaks of specific biomolecules you know. And we do all of that, but in the interest of time, we're going to move on. And back we go, switching gears, project number 2. All right, so first project, we look at the bottom left, and second project, we look at the top right. And the idea is here, instead of looking micro, right, at one cell line, just a bunch of cells in extreme detail, we go macro. So we try to make a model for all cells in the world, all at once. We'll have to compromise a little bit on detail, obviously. So what's the kind of equivalent for cells of the tree of life? Now, when you think about evolutionary biology, for a long time, people ask themselves this question: what is a species? And that is very hotly debated to this day in those departments. In my personal, very biased opinion, that's maybe not such a useful question to ask. And maybe this is a slightly better question to ask, which is that as all models, it's never going to be perfect, but is it useful for something? And obviously, species are useful as a model because they help you learn a lot of evolutionary biology really quickly. And I'll show you how. You can go to the Australian Museum at 9 a.m. at opening time with your 2-year-old daughter and put her in front of the tree of life, and she'll be like, wow. And you're like, all right, it hardly gets easier and quicker than this. That's why they put it in the museum. So the question is whether you can do something similar for cells. So instead of asking what's a cell type, which is very hotly debated all the time in the community, maybe you can ask what is a cell type model useful for? And of course, the answer now we know is to learn a lot of cell biology easily and quickly. So it has to be easy and quick. So that's why we developed this project called the cellular approximations. And Yingshu, the software engineer behind this, is here with you. So bother her if something is wrong, not me. Thank you so much. She also made a YouTube channel. So if you want, you guys can watch the tutorials, which are very nice, and like and subscribe. And if she doesn't push out enough updates, you can bug her and say, I want a new video this week or something, and I'll support you. So let me show you how that works. So this is the website, and it contains all cells on Earth. So you can say things like what species are available. I mean, it's a database, so only so and so many species have been sequenced at the single cell level, but we have most of them, I think 90%. And say maybe you're like, huh, okay, fly is interesting. So you go explore fly. Okay, so this is a fly. These are all the organs of the fly that got single cell sequenced. And as you can see, there's a bug because you cannot see the full name of the organ. Anyway, you can click on one of them and then go to, I don't know, tracheocyte. And then when you click there, it prompts a question for the chatbot. And then you can go there and it shows you marker genes for tracheocytes in the fly trachea. And you can zoom in and it shows you cell states. And you can see each cell state is composed of cell types and so on. And you can do a lot more things. I'll show you another one just for fun, in which you can look at, let's see. You can look at these, for instance. So you can look at which cells are present in which organ. Right, this is really like museum level, like children can use this, right? And there's even one that I can't remember how to type it now, but it shows you cells that are shared across different organisms and in which organ they are. So that's also fun. All right, I don't want to keep you here forever, so let's go. I pressed the button I shouldn't have. I did.
This is using the
Good idea, good idea. It says it's open, but it's not really open.
It's populated screen, so this so we just
Oh, okay. I see. Okay, thank you. There you go. That's the last slide. I don't want to keep you here forever. These are all projects I don't talk about today. Thanks, fortunately for you. All right, so I just want to give you kind of a quick sense of the software engineering behind this. You need to make this quick and easy. As you've seen, the answer come within a second or a second and a half. So you need to simplify all these big single cell data sets in a way that they become quick. We have a few ways to do that. You need to make it machine queryable if people want to train machine learning on it, and then you have to make it browsable by children. And so you need a chatbot and a place where they can go click. All right, and that summarizes the talk. I've shown you two little adventures, a little bit maybe odd ones compared to the mainstream. about cellular diversity. One is HyperSeq, a way to look at extreme detail of individual cells and connect imaging with sequencing. And another is class approximations, which is a museum level, I suppose, simplification of all cells in the world in one place. And thank you for listening, and I'll be happy to answer any questions.